Hello, science enthusiasts, and welcome back to Biotic World, the ultimate destination for deep dives into the world of biology. Today, we are exploring a cutting edge technology that's transforming the future of agriculture, horticulture, and conservation plant tissue culture. This powerful technique isn't just a tool for growing plants, it's revolutionizing the way we propagate, preserve, and even genetically enhance plant species. Buckle up, because today we are diving into the advantages of plant tissue culture and its potential to shape the future of plant science. To kick things off, let's understand what plant tissue culture really is. In its most basic form, it's the technique of growing plant cells, tissues, or organs in an artificial environment using a nutrient medium. These tissues, when exposed to the right conditions, can regenerate into entire plants. This technique is not just for research, it's widely used in the production of genetically identical plants, and the advantages it offers are immense, especially in terms of genetic conservation and crop improvement. Let's get into the advanced advantages of plant tissue culture. We'll go beyond the basics and explore how these benefits are reshaping modern agriculture and biotechnology. First is about the clonal propagation and genetic uniformity. One of the most fundamental benefits of plant tissue culture is clonal propagation, where a single plant can produce genetically identical offspring. This is essential for commercial agriculture and horticulture as it ensures uniformity in traits like flower color, fruit size, or disease resistance. Let's consider potato farming. Through tissue culture, farmers can produce large quantities of potato plants with identical genetic profiles, leading to consistent crop yields, uniform quality, and higher profitability. Second is about the elimination of pathogens and disease-free plants. Plant tissue culture offers a significant advantage in producing disease-free plants. Traditional methods of propagation such as seeds or cuttings often carry plants disease and pathogens. Tissue culture helps mitigate these by growing plant material in a sterile environment. Through techniques like meristem culture, where a small piece of tissue is taken from the tip of a plant's growing shoot, scientists can produce clean plants that are free from common diseases like tobacco mosaic virus or bacterial wilt. Next is about the conservation of rare and endangered species. Plant tissue culture plays a crucial role in the conservation of endangered plant species. Certain species, particularly rare or threatened plants, can't be propagated using traditional methods due to their limited reproduction capacity or complex growing requirement. With tissue culture, genetic material from these plants can be preserved and propagated in vitro. This not only aids in conservation but also allows for genetic banking, where the genetic material is preserved for future use, ensuring the survival of species that might otherwise be lost to extinction. Next is about the exponential plant production. One of the key advantages of plant tissue culture is its ability to produce large quantities of plant in a short time frame. Traditional propagation methods can take months or even years to produce a significant number of plants, but tissue culture can multiply plants rapidly, sometimes in just a few weeks. This is particularly important for high demand crops like bananas, grapes, and flowers. The process of micropropagation allows for the rapid scaling of production without sacrificing plant quality. Next is about the genetic improvement through clonal variation. Tissue culture is not just about cloning, it's also a powerful tool for genetic improvement through a process called clonal variation. Genetic changes can occur during the tissue culture process. These variations can be selected for desirable traits such as enhanced disease resistance, drought tolerance, or increased yield. It's a way of inducing mutations at a controlled level. which can lead to the development of plants with better adaptability to environmental stressors, making them more sustainable in the face of climate change. Next is about the customized plant breeding with genetic engineering. Plant tissue culture also serves as a critical technique in genetic engineering by combining tissue culture with tools like CRISPR-Cas9 or gene guns. Scientists can insert specific genes into plant cells, creating genetically modified plants with desirable traits. Whether it's creating plants resistant to pests or developing varieties that can thrive in saline soil, tissue culture allows for precise genetic modifications that can be reproduced efficiently. Next is about the space and resource efficiency. Tissue culture offers a solution for space efficiency, which is becoming increasingly important as urbanization and climate change put pressure on arable land. Unlike field based growing methods that require large expanses of land, tissue culture can take place in compact lab environments where plants grow in small containers under controlled conditions. This allows for vertical farming or urban agriculture, where plants can be grown on multiple levels, conserving space and resources. Additionally, tissue culture can use less water and fertilizer compared to traditional methods, making it more environmentally friendly. Next is about year round production. One significant limitation of traditional farming is the dependency on seasonal cycles. With tissue culture, plants can be grown year round, regardless of external environmental conditions. This is particularly beneficial in greenhouse farming and climate controlled production systems, where crops can be continuously produced without the interruption of seasonal weather. This ensures a steady supply of plants, reducing the risk of market shortages and increasing food security. Beyond traditional agriculture, plant tissue culture has revolutionized the field of biotechnology, and it's being used for a variety of applications that are reshaping the future. Certain plants are used for the production of medicinal compounds, such as Taxol from the yew tree. Through tissue culture, these plants can be grown in controlled environment to produce a secondary metabolites, which are then harvested for pharmaceutical use. This reduces the pressure on wild population and ensures the sustainability of medical plant resources. This is about the pharmaceutical production. Next, we are talking about the transgenic plants. Plant tissue culture plays a pivotal role in the development of transgenic plants, which have been genetically modified to express specific traits. This includes improving crop yields, enhanced resistance to pests and diseases, and even biofortification, like producing plants with higher nutritional value. As we have explored today, the advantages of plant tissue culture go far beyond simple plant propagation. From rapid multiplication and disease free production to genetic improvement and conservation efforts, plant tissue culture is a cornerstone of modern agriculture and biotechnology. It's enabling us to grow smarter, more sustainable, and more resilient plants, paving the way for a greener and more food secure future. If you found this deep dive into plant tissue culture fascinating, don't forget to hit the like button, subscribe to Biotic World, and ring the bell icon so you never miss another exciting video. Drop your thoughts and questions in the comment section. I'd love to hear what you think about the revolutionary impact of plant tissue culture on the future of farming and conservation. Until the next time, keep exploring, stay curious, and continue to unlock the secrets of biological world.